Okay, day six. Day six. We are here. We're live. We're still learning. We're still learning. I changed the microphone so when I cough, like something with my throat, I'm gonna do that strategy. 1-1 should be better. And we stopped on a question number 21. You mentioned something interesting. You said viruses can have DNA. Can viruses? Today I want to. What we're gonna do today? Today I feel like today I want to learn about T cells. T cells. T cells, the ones that are probably gonna rule. Let me minimize all that stuff. And I also need to open another thread, most likely. New cloud. And select a project. Blah, blah, blah. I'm still using a browser for now. But eventually we're gonna switch to assistants like on-demand assistant. That's just listening everything what we're saying, or particular I saying, and then double check, like reality check on me. It's pretty straightforward to implement. It just I'm not at this stage. So I actually want to know this. Remember how I was thrown away by respiration and aerobic and anaerobic? Okay, recording a video. Oh, I need to click save. 1 second, guys. Couple more. Save button. Okay, anyway. Okay, here's the thing. There is two way how we can break down the sugar. Aerobic and anaerobic respiration. With aerobic one, when we use the oxygen, there is a 36, I believe, right? Approximately ATPs created in mitochondria from 36 till 38. And anaerobic respiration, only 2 ATPs per glucose. So the question is it possible to put our organism in a state of anaerobic respiration for a long time? And long time, I mean like a few days. So it's kind of hibernation, but just switch our cells into anaerobic respiration so it can burn more sugar because it's need ATPs, right, for everything. So it will be forced to burn more sugar. Sorry, not sugar. But it's fast and doesn't need oxygen. So it kicks when demand exceeded. Yeah, glucose. My guess is not, because our brain needs way more energy than the rate of 2 ATPs per glucose. Right? Like, what's the speed of breakdown and producing ATP per cell on aerobic respiration versus anaerobic respiration? I think, let's say, like, our consumptions probably need like 50,000 ATPs per second. Maybe even more. Probably more, way more. But anaerobic can produce only like half of it. Lactate is a real bottleneck, not ATP math. Okay, I see. Your intuition is right, but here's the reason coming. Let me share. Can you force the body into sustained anaerobic respiration to burn more glucose? No, and for reason worse than the brain needs more ATPs. Interesting. A few second problem. Lactate is the real button. So we don't have mechanism to remove lactates fast enough. Anaerobic is mammals doesn't just stop at 2 ATP, it produces lactate. And positive, that's not a proton, is it? As byproduct, because like I print this one on a day one, and it's like needed me everything, and it needed me every single day. This is so cool. Like, why I was showing, because it was H plus there as well. As a byproduct, because NADH has nowhere to dump its electrons without oxygen. Think of NAD plus as a limited pool of electrons carrier. Objects that glucose needs to borrow and return. In aerobic mode, the mitochondria recycles them via the electron transport chain. I just showed that picture, this one. In anaerobic mode, you generate NAD plus by converting pyruvate lactate. That works for a sprint, but over hours you accumulate lactate faster than the liver can clear it, via the Cori cycle, with itself costs 6 ATP per glucose recycled. Oh, so I need a better lactate clearance. So you actually do net negative system wide. Okay, brain has a harder aerobic dependency. 20% of your resting ATP budget goes to the brain. I knew that. Called it. A new brain uses a lot of energy, yep. The brain and neurons basically cannot run on glucolysis alone. They lack the glucolytic capacity to meet demand, even if substrate were unlimited. Substrate was unlimited. Under hypoxia, consciousness goes in 10 seconds. Under hypoxia, consciousness goes in 10 seconds. So basically, 10 seconds in hypoxia, and I'm assuming that's without oxygen, right? Oxygen starvation. I need to listen to this recording and see if it helps with my coughing, because I don't know what's happening. 10 seconds in irreversible damage starts in 5 minutes, so few days of anaerobic mode isn't survivable regardless of glucose supply. Interesting. Your premise has the economics backwards. You're framing aerobic mode as a burns more glucose, which is good. But the organism doesn't want to burn glucose. It wants ATP. Glucose is a fuel. ATP is a product. Yeah, I know it doesn't want to burn glucose, but I do want to burn glucose. Oh no, I do want to burn fat. Switching to 2 ATP pathway to burn. So is it possible to develop some kind of mechanism to get rid of lactate on a cellular level and dump it through the sweat, let's say? And that's probably will work on a regular tissues, but in a brain, yeah, interesting. Can we synthetically produce ATP and plug it into the brain? That's 2 questions for you. And one more. Yeah, I definitely need the ATP demand drops and hibernation. Let's also consider that. And also, what is the pathway of ketosis? Who convert fat into the energy, into the ATP? I'm assuming ketones, but how do they do that? We're probably gonna YouTube it really quick. Dr. Huberman, he's the man. I don't need diets, I need a molecular structure. What's the pathway? Let's see if I spell any of those keywords correct. Ketosis. Oh yeah, that might be it.
But did you know that you can fuel cold cytoplasm with the process glycolysis, where glucose is converted into pyruvate? The pyruvate then moves into the mitochondria.
Glycolysis.
Glucose starts in the cell's cytoplasm with the
Cellular respiration.
process glycolysis, where glucose is converted into pyruvate. The pyruvate then moves into the mitochondria, where it's oxidized to acetyl coenzyme A by the enzyme complex pyruvate dehydrogenase. Acetyl-CoA is the starting material for the citric acid cycle, which reduces electron carriers ready for use in the electron transport chain where the bulk of ATP is made. ATP, or adenosine triphosphate, is the molecule that provides energy to all the cells in the body. If more carbohydrates are consumed than needed, the excess glucose will first be stored as glycogen in the liver and muscles. Then, when those stores are full, excess glucose will be converted to fatty acids and stored in adipose tissue. Using fats for energy. When dietary carbohydrates are sufficiently restricted, such as when fasting or on a low carbohydrate diet, the body will start to make ketones using
Here we go. Cool animation, by the way. What's this logo?
exogenous fat, which is the fat you eat, or endogenous fat, which is the fat stored in your adipose tissue.
Mm-hmm.
There are three types of ketone bodies: acetoacetate, acetone, and beta-hydroxybutyrate.
Acetone, acetate. Okay, and this one is pretty devastating. When it goes to the skin, it will give you like red marks. You basically burn skin by acetone. Is it same acetone that you use to remove not nails, but paint from your nails?
Butyrate. These molecules are commonly referred to as ketones, but chemistry enthusiasts might notice that only the first two are in fact true ketones.
Okay, yeah. I'm not a chemistry enthusiast, but I have to become one. This one extra H, this one completely off.
While the beta-hydroxybutyrate is a carboxylic acid. The process of making ketones is called ketogenesis, and it occurs mainly in the mitochondria of the liver.
Okay, so ketones made in liver mainly, but also by mitochondria.
Fatty acid molecules are enzymatically broken down via beta oxidation to form acetyl-CoA. Then, through a series of reactions, acetyl-CoA is converted to 3-hydroxy-3-methyl-gluteryl-CoA, which goes on to produce the first ketone body, acetoacetate. The acetoacetate then produces the two other ketone bodies, acetone and beta-hydroxybutyrate. The body cannot metabolize acetone, so it is expelled via the breath.
Yeah, I noticed that. The people who is in the ketosis, I noticed their breath smell differently. It smell like a hospital. Okay, so interesting. So acetone it's a byproduct that we cannot metabolize. So it's actually like goes out of body with breathing and also through the sweat. But I thought acetone is ketones that's used for breaking down fat.
Acetate then produces the two other ketone bodies, acetone and beta-hydroxybutyrate. The body cannot metabolize acetone, so it is expelled via the breath. The two remaining ketones, acetoacetate and beta-hydroxybutyrate, then leave the liver and are transported via the blood to other tissues throughout the body, including muscles and the brain.
Okay.
Unlike fatty acids,
so it's only into brain and muscles. Okay.
Ketones are able to cross the blood-brain barrier to direct.
Okay, so it can go to brain-blood barrier. I heard about it.
Ketones are able to cross the blood-brain barrier, acetate and beta-hydroxybutyrate, then leave the liver and are transported via the blood to other tissues throughout the body, including muscles and the brain. Unlike fatty acids,
that's what I was like. Okay, unlike fatty acids cannot go to brain, therefore our brain not getting fat. Yep. All clear so far. By the way, Fire Films, good job.
Ketones are able to cross the blood-brain barrier to directly provide energy for the brain, just like glucose. Once in their target cells, the ketones are converted.
So it goes to the brain, it going to the cells of brain. Inside of cells of brain there is mitochondria, and now still mitochondria produces the energy. It's just not from the glucose. Now it's produced from the ketones.
Converted back into acetyl-CoA, ready for use in the citric acid cycle to continue the supply of NADH and FADH2 to the electron transport chain to produce ATP. It can take several days to weeks for the body to adjust to using fat for energy, but there are many benefits to reducing carbohydrate consumption. Emerging research indicates keto adaptation has transformative disease fighting and performance enhancing effects. So why not give it a go?
In this dirty biochemistry lesson, we're going to continue our discussion and talk about ketone body synthesis. Ketone body synthesis acids in the liver. It typically occurs when glycogenolysis, ketone body synthesis, and so everything that you see here
TCA cycle, fatty acids
in blue
oxygenation.
is co-production of shown here in start with acetyl-CoA, and specifically prevents us from getting more glucose from gluconeogenesis, but it prevents the TCA cycle from working.
Great presentation, but I lost. Okay, let's see what's this one says. Good question. Let me take them in order. Lactate disposal via sweat or other routes, like we do disposal of acetone in the ketosis, right? Clever idea, but the numbers kill it. At rest, you burning 2000 kilocalories per day. Maybe. When I sit and I burn less. Call it 150 grams of glucose equivalent. Really? If you run the anaerobically, you'd produce 150 grams of lactate per day. And lactate in sweat is present only in trace amount. Millimolar range. Millimole, I guess, right? Similar to blood. Sweat glands don't concentrate it. They passively leak what's in interstitial fluid. To dump 150 grams per day via sweat, you'd need to sweat many liters and actively pump lactate against its gradient, which would itself cause ATP you don't have. Okay, fair enough. The kidneys are better out. They already excrete small amounts, but they cap out around a few grams per day. Okay. Before the filtration machinery saturates. The liver co-recycle handles the real volume, but as I mentioned, it costs 6 ATP to recycle one lactase back to glucose, so you're running a net negative energy column. Yeah. You could imagine engineering system lactate exporter. Bacteria have lactate dehydrogenase that work differently, and some organisms handle lactate loads we can't. But even if you solve the export problem, you're still stuck with the charge balance problem. Lactate leaves as a lactate minus. Oh, I see. The charge, right? And you have to co-export H plus, or you alkalinize the cell. So the pH. I'm not sure if pH balance is relative here. So, but you cannot make it alkalize because the cell will just die. Does this? The bottleneck is downstream where the lactase goes. Something has to oxidize it back to pyruvate. So we cannot just invent different routes. We have to always close the loop. Close the loop in biology, close the loop in the AI agents, always close the loop. If you don't have a loop that closes, then it's not a real system. You could imagine that lactates have lactate work different, and some organisms handle lactate loads we can. Yeah, I read that. Export plan your sticks charging the MCT monomer transporter family already does this couple export. MCT oil, right? That's what it's come for. It's not the bottleneck. The bottleneck is downstream where the lactase goes. Something has to oxidize it back to completely. And yes, your brain intuition is correct, but for subtle reasons, then brain is different tissue. The blood-brain barrier is selectively permeable, and neurons actually import lactate from astrocytes as fuel. Okay, I'm totally lost in the synthetic ATP infusion to the brain. No, and this one fails. And this one fails at the physics layer, not the engineering layer. ATP doesn't cross cell membranes. It's a large, highly charged molecule. For negative charges at physiological pH, membranes are specifically designed to keep it in. Cells don't have ATP imported because no cells in evolutionary history has ever needed one. ATP is made in-house. If you IV'd ATP, it would get hydrolyzed in the bloodstream within seconds by ETA ATPases enzyme on vessel's walls that break it down. Nerves cross the blood-brain barrier even if it survives. Never enter nerves even if it crosses the BBB. Or BBB, it's a brain-blood barrier. Trigger inflammation, yes. Lowering ATP demand and plasma. Now this is interesting version of your original question because it's the path biology actually uses. Hibernation, ground squirrel, bears, some bats drop metabolism rate by 98%. Body temp fall from 37 near freezing in depth hibernation. Heart rate goes from 200 plus. Wow, and critically they stay aerobic the whole time, just very slowly. The trick isn't switching pathway, it's lowering the clock. Answering the ketosis, the pipeline, lipid, yeah, we saw this. The brain dips ketosis can get 60-70% of energy from ketones, with the remaining 20 of still requiring glucose, which the liver provides via gluconeogenesis, making glucose from amino acids and glycerol. It can never get 100% ketone. Certain neuronal function generally requires glucose. Ketosis is evolution's answer to your original question. In a way, how do you keep the brain fueled when glucose is scarce? Not by switching to anaerobic respiration, but by inventing a new aerobic fuel the brain can import. Love it, let's go. Oh, the T cells. Actually, I want to... T cells. I feel like this is too simplified. 9 minutes. Yeah, I want to watch this one.
And humoral immunity as well as cell-mediated immunity. And based on the textbooks I've read, a lot of these texts just make a huge complicated mess out of this topic. So I'm gonna try to break it down Barney style. I'm trying to make it as easy as possible. So I will be bringing up an illustration from our Medical Surgical Edition 2.0 deck. If you are following along with cards, this illustration is on card number 4. Okay, let's go through the steps.
Okay, I need to get those cards. Antibodies, antigens, B cells. I know all that now. Memory cells, which is B cells. Plasma cells. I don't know what is this. Interesting. Helper T cell. I need those cards. Where can I get those cards? Head over to interactive study guide and anytime find out. Access CAM courses or helper. Oh, that's a lot of pointers. Way more to master medical surgery nursing. Games, digital resources, perks. I need card number 4 or 24. Oh, that's the cards. Med surgery. 99 bucks. The study knowledge is expensive. I mean, these days probably knowledge is cheap, but everything you need to know available on the internet, I guess. But this is a way to learn it.
Of humoral immunity using this illustration. So first of all, we have an antigen that will bind to an antibody on a B cell. The B cell will engulf the antigen and present it on the surface of the cell by MHC2 proteins. Then this helper T cell here in green will bind to the complex and release cytokines, which are basically like chemical messengers. This will induce this B cell to rapidly divide into plasma cells as well as memory cells. These plasma cells will produce and release antibodies that will work against this specific antigen. The memory cells will become sensitized, but they will not function until the next exposure to the antigen. At that time, when they are re-exposed, they will mount a large and rapid response to that antigen. So they're like, yeah, I'm waiting for you. So they're like, yeah, I'm waiting for you. You come around again and you're going to be in trouble. So
I'm not sure, like, so releasing helper T cells, releasing cytokinase, the signal, right? So why don't, is it same cytokinase for all? For all type of antigens? Same signal for all type of antigen, just saying like, hey, make more B cells. But what kind of B cells, right? Because each B cells has a different antibody and different memory of antibodies that's matched onto antigen.
They're ready to go next time the body encounters that same antigen. So then we have these antibodies here, which will bind to the antigen, and with that binding, a reaction takes place, and that triggers neutralization and destruction of those antigens. All right, now that we've gone through the steps of humoral,
I don't know. Let's go with this one.
Steps in this video will cover the response of the immune system to viral and bacterial infection, the difference between T cells and B cells, and provide an explanation of the adaptive immune system. Number 1. There are 2 branches to the immune system.
Yep.
The 2 branches of the immune system can go by the names specific and nonspecific, innate and adaptive.
That's what I learned, innate and adaptive. Innate, it's mediated by macrophages, and adaptive, that's for B cells and T cells. Oh, so the B cells also engulf like macrophages, right? Is the B cell is a type of macrophages?
As well as some other lesser common names. For the purposes of this video, is the immediate and quickest response to an infection. However, it is not very specific. It often is not enough of a response to contain the
All these are macrophages, right? Natural killer cell. Macrophage. Dendritic cell, which is type of macrophage.
infection. This is the reason for the adaptive immune response. The remainder of this video will cover the adaptive immune response. For more information on the innate immune response, see the video on inflammation on this channel. Number 2. B cells and T cells are the fundamental cells of the adaptive immune response. The adaptive immune response consists of humoral-mediated immunity and cell-mediated immunity.
So the adaptive split into two parts as well. Humoral mediate immunity and cell mediate immunity.
Humoral-mediated immunity is the name for immunity that is provided by B cells.
B cell antibodies, T cells, T cell, CD4 plus, CD8 plus, YAT cell, natural killer T cell. What's this difference?
Cell-mediated immunity is the name for the immunity provided by T cells. Humoral is Old English, referring to the humors of the body, meaning the fluids of the body. B cells are able to provide immunity in the humors, or fluids of the body, because B cells secrete antibodies to circulate throughout the blood and other.
Oh, I see. So B cells, they in the fluids, like in the blood, right? And I saw an animation how B cells, maybe it was B cell or maybe it was macrophage, going from the bloodstream, sliding between cells of the blood vessels and going into the tissue itself.
Fluids of the body. If an infection has reached the inside of a cell, humoral-mediated immunity will not be effective. This is why cell-mediated immunity exists. T cells protect the body from infections that have reached the inside of cells. Thus,
Oh, I got it. So B cells to kill everything outside of cell membrane, whatever what's in the fluid. And T cells to kill something what's inside of this. Now I have a question now. Hold on. So my question is, immediate immune system use macrophages to attack pathogens and eat them alive using cell types called macrophages. And looks like B cell doing same stuff, but also release antibodies and release some kind of signaling for the T cells to release even more signal to create more B cells. But anyway, my question is, I'm studying B cells and macrophages and they look pretty much identical to me. Is B cell is a type of macrophage? If yes, then what's the difference? If not, then what's the difference between macrophages that attack pathogen right away and B cells that's actually more adaptive immune system branch? I think I asked it well. Good question, and the confusion is understandable because they do have overlapping behavior. Both can engulf material. Yep. Both present antigens. Yes, both are part of coordinated immune response, but they fundamentally different cell types with different lineages and different jobs. Let me untangle this. No, B cells are not macrophages. Okay, they distinct cell lineages that diverge earlier in hematopoiesis, blood cell development. Think of it like this. Both descend from the same stem cells in the bone marrow, but they branch at very early fork. Okay. Initially, all blood cells come from hematopoietic stem cells. HSCS in bone marrow. The first major fork split into two lineages. I need a video animation for this. If you know a good one, link me. Myeloid lineage, macrophages, neutrophils. That's a cool name. Neutrophils. Dendritic cells, mast cells, red blood cells, platelets. How is the red blood cells? Where's the red blood cells getting energy if they don't have mitochondria? That's my next question. Where does red blood cells get the energy if they don't have mitochondria? Wow, that's a very big answer. Immune oxygen transport clothing branch lymphoid lineage, B cells, T cells, and K cells. This is the adaptive immunity branch. Mostly NK cells are an exception. They are innate. Can you imagine all this branching happened by evolution? Like, I can't. This is way too complex. Like, looks like someone designed that system. Macrophages and B cells are different branches. Okay, I got it. The branches is different. They share ancestor. The specific similarity you are noticing. Both in Gulf. Both cells can. I remember I add that word into my vocabulary, but I never went through the vocabulary learning, which I should do.
Phagocytos.
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 Phagocytos material, and both can present antigen via MCH class 2 molecule. This real or and it's what makes them look similar in introductory material. But the purpose of this behavior is different and the machinery is different. Macrophage is the generalist garbage collector, first responder. Innate immune system. Pattern recognition, they recognize broad molecular signature common to many pathogens, LPS and gram-negative bacteria, flagellin, double-stranded RNA, etc. via receptors called PRRs, pattern recognition receptor. Oh, I love this. Most famously, toll-like receptor, engulfing anything that looks foreign or dead. Pathogen denies cell debris. Pathogen dying, cell debris, and regular debris. Deadjusted in lysosomes and either present fragments to MCV or just destroy it and move on. So when it's presented fragments on MHC2, then B cells comes, sorry, not B cells. The T cells probably comes and send a signal, hey, we need more of this. Releases cytokinesis to recruit other immune system. See, I was wrong. I thought T cells or B cells comes and releases cytokinesis, but it's actually same macrophage doing it. Live in tissue long term. Really? Live in tissues long term. Alveolar macrophages in lungs, Kupffer cells in liver. Microglia in brain, osteocala in bone. The residence genitors with weapons. Okay. Key property, no memory, no specificity. They recognize bacteria, not the specific strain of E. coli I saw last month. Okay. B cells, the special antibody factories. Lymphoid and adaptive immune system. When B cells bear the binds its specific antigen, it internalizes it, processes it, presented fragment on MCH2 to helper T cells, and if it gets the right T cell signal, activates proliferates clonal expansion and different. Oh my god, that's so complicated. Plasma cells. Pure antibody factories pumping out soluble versions of that B cells BCR into the blood. So B cells live in fluids. Memory B cells, long-lived cells that remember this antigen and responds faster the next time. Key private specificity. Memory. Amplification. One activated B cells, millions of plasma cells secreting antibodies. Okay. Amplification of signal, memory, and specificity. It could just list that. The pathologist thinks why both do it but differently. Programming analogy. Macrophages run for item in environment if looks for origin item, engulf and destroy item. AB cells run if matches cell-specific receptor, engulf and present two cells item. Await activation signal, same and engulf. Subroutine, completely different control flow around. Love it. I actually love this. Like, if you already expert in one area niche, to learn another, it's pretty simple by just reference what you already know. And that's how memory forms, right? It's attach itself to the analogy. Antigen presents the or other overlap. Why they look similar in textbook? Both are roughly round. I mean, they all round molecular cells under microscope. Both do antigen presentation. Both are part of the immune response. If you're looking at cartoons of self-is pathogens, they look identical. The difference are the receptors they express, which you can see without staining. Oh, you can see without staining. The transcription factor driving them and the downstream behaviors. Macrophages are the tissue resident infantry. Generalist, always ready, no memory. B cells are specialist R&D labs. One lab per possible threat. Slow to activate the first time, but capable of producing precision-guided munitions, antibodies in huge volume without instructional memory or for the next time. Yep. Great question. So now we're going to back to the question, where do red blood cells get energy if they don't have mitochondria? That's my question for day number 6. And as always, a great question. And the answer is elegant. Red blood cells run purely on anaerobic glycogen. Here we go. We connected the dots. So it doesn't need mitochondria because it produces its own ATPs by anaerobic glycolysis. Yep. They are the one cell type in your body that permanently lives in the metabolic state you were asking about in your first question. We connected the dots. The setup: mature red blood cells, erythrocytes, have no mitochondria, no nucleus, no ribosome. No nucleus. Where does it store DNA? No ribosomes, no ER. During maturation, they eject all of it. Oh, they use it to grow and then like, bye bye. What's left is essentially membrane bound bag of hemoglobin with just enough cytoplasmic enzymes to keep itself alive for 120 days. Oh wow. So we regenerate blood every 120 days. This is wild design choice and it's deliberate. It is a wild design choice. That's so cool. It's like kind of sending a rocket to the space and jettison everything that you don't need anymore, right? You survive for like 120 days and then it just go away from the orbit or like fall back to the atmosphere and burn. This is a wild design choice indeed, and it's deliberate. Consider an RBC's job is to transport O2 from lungs to tissue. If it had mitochondria, those mitochondria would consume some of the O2 it's carrying. True. The cargo would be eating itself in transit. By throwing out the mitochondria, the RBC becomes a pure O2 shuttle that doesn't tap its own payload. Wow. How they make ATP? Yeah, we already know that. Probably. Membrane flexibility, keeping hemoglobin reduced. Iron occasionally oxidizes from F2 plus to F3 plus. Methemoglobin, which can't bind O2. Reducing it back requires NADH from glycolysis, powering. Day 6. Like, this is different language. Why is this working? Okay, I need to finish those questions, and the reason why, because then I will be given a prompt with my basis, like, and AI can explain me in my language, so I can slowly gain the traction of vocabulary and understanding, but not just be throwing the pages of things that not I just don't know, the things that I cannot even read. Okay, let's go. You mentioned something interesting. You said viruses can have DNA. Can viruses also have RNA instead of DNA? And you name one example of a RNA virus. Yeah. Viruses actually don't have DNA. They only have RNA. So they have a chunk of DNA. I remember from yesterday's session, we were talking about HIV. And HIV actually lives in cells. That was interesting to me. Give me more information about like how does it inject itself into our own DNA? And what happens if the person have kids? Would that be passed to the kids? Probably not. All right, it will not, because it's not in the sperm. Yeah, I don't know the answer to that question. I think it only has RNA. level up.
Thus, by providing immunity in the fluids of the body through B cells, and by providing immunity from infected cells of the body through T cells, the adaptive immune response is able to provide immunity throughout the human body. Number 3. B cells and T cells contain infections in different ways. In immunology, putting an end to an infection or bringing an infection under control is often referred to as containing an infection. While B cells and T cells are both components of the adaptive immune response, they contain infections in different ways. B cells produce and secrete antibodies. Antibodies are proteins that are able to bind to bacteria and viruses. Antibodies binding bacterial cells or viruses prevents infections from spreading. The antibodies block.
That's what's confusing to me. Like, they showing that B cells have to be present here, but it's not true, right? It's release antibodies, and then antibodies just kind of surrounding pathogen. The B cells might be like kilometer away.
Block the bacteria from growing colonies, and in the case of viruses, the antibodies bind to the viruses and prevent the viruses from infecting cells. Any disease-causing agent, such as a virus, can be referred to as a pathogen.
Any disease-causing agent, such as virus, pathogen. Any disease, that's interesting. Any disease-causing agent.
The process of antibodies binding a pathogen to contain the infection is known as opsonization. Opsonization by antibodies not only prevents the spread of infection, but marks the bacteria or viruses for destruction.
Okay. Yeah, that's what okay. So sometimes B cell actually do it themselves, sometimes they release. Okay.
Phagocytic cells circulating in the blood and fluid.
Okay, they lock into antibodies, understand it, and eat it.
Surrounding the tissues of the body can detect the presence of antibodies bound to pathogens and then engulf and destroy the opsonized bacteria or viruses. These phagocytic cells are not B cells or T cells, and
why they showing different one? Because they live in different tissue. Now I understand. So I need to know which one to live where.
are part of the innate immune response. So while the process of opsonization is part of the adaptive immune response, the actual destruction of pathogens involves the innate immune response.
Okay, so they just surround it, but then innate one still eating it, not the B cells. So the card that I show was super misleading. It was showing that B cell eating it.
Response. Therefore, both branches of the immune system must work together for a complete immune response to infection. Further detail on this phagocytic process is provided in the video on this channel on inflammation. When a cell becomes infected and cell-mediated immunity is needed, T cells respond by
Cell-mediated immunity.
killing the infected cell. There are numerous needed inflammation. When a cell becomes infected and cell-mediated immunity is needed, T cells respond by killing the infected cell. There are numerous types of T cells.
Oh, I see. So because macrophages cannot eat our own cells because they're not foreign, the recognition doesn't work. But when our cell become infected, the T cell saying like, too bad, I'm gonna kill you.
Cell-mediated immunity is largely dependent on a class of T cells known as CD8 cytotoxic T cells. The CD8 cytotoxic T cells are called cytotoxic because they are able to kill other cells. When a cell becomes infected with a virus, a CD8 cytotoxic T cell responds by killing the cell infected with the virus. Since viruses replicate inside of cells, killing the cell infected with the virus stops the ability of the virus to replicate. Additionally, while often not sufficient to rid the body of cancer, CD8 cytotoxic T cells are also able to kill cancer cells when they detect cells of the human body that have become cancerous. Thus, while CD8 cytotoxic T cells are killing cells of the human body, whether virus-infected or cancerous cells, this can actually preserve health overall in the body because the killing of the infected cells can stop the spread of the virus or cancer. Cell particles and viral proteins that are released into the fluids of the body after the infected cell is killed can then be destroyed by phagocytic cells, just as is the case with opsonization. To summarize adaptive immunity, B cells respond by releasing antibodies for opsonization of
now I know
pathogens,
what's
resulting
B
in the
cells and T
destruction of the
cells.
pathogens.
Difference.
T cells respond to infected cells by killing the infected cells. Specificity is the basis of adaptive immunity. As previously mentioned, adaptive immunity is often a more effective response at containing an infection than innate immunity. This is because adaptive immunity is specific. What this means is that adaptive immunity is focused on one specific type of pathogen. For example, if a person is infected with a strain of the flu, the antibodies that protect from that infection are specific only to that one strain of the flu. If those same antibodies that stopped the spread of this one strain of the flu were then exposed to a common cold virus or a streptococcal strain of bacteria, the antibodies would not be able to bind and would not at all be effective in containing the infection. This is due to the specificity of the antibody binding site. The portion of the antibody that binds to a bacterial cell or a virus is known as the binding site. The binding site on an antibody has a specific shape. This shape determines what an antibody is able to bind to. If an antibody comes across
And it's made from protein, right? Both of them. So we basically need to mirror the shape, but opposite. It's like negative shape.
a bacterial cell or virus, and the shape of the binding site on the antibody is not a good fit for the bacterial cell or virus, the antibody is not able to bind and will simply continue to pass through the fluids of the body. One B cell can secrete one type of antibody, meaning the antibodies will all have the same shape to their binding sites, resulting in the same specificity for one type of bacteria or virus. However, since all antibodies secreted from this B cell will have the same shape binding site, this means none of the antibodies secreted by this B cell will be effective at stopping a different kind of pathogen than the ones these antibodies are able to bind. For this reason, numerous B cells are needed for
What happened with the antibodies left over? Do they been eaten by macrophages?
proper immunity. While one B cell may secrete antibodies with one binding site, another B cell will secrete antibodies with a different binding site, and so on, demonstrating the need for multiple B cells as part of the immune system. In the case of T cells, the T cell receptor is the specific site.
T cell, B cell, lymphoma, cancer cell.
Just as is the case with the binding site of antibodies, the shape of the T cell receptor on T cells is what determines which infected cells a T cell will bind to. If a T cell comes across a cell infected with a virus, and the T cell receptor is able to bind the infected cell, the T cell will become activated and release cytotoxic molecules that can kill the infected cell. The same T cell, however, may come across a cell infected with a different virus, and the T cell is unable to bind, and no cytotoxic molecules will be released. As is the case with antibodies, T cells are specific to being able to respond to only one kind of virus.
Okay.
There are numerous T cells, however, and an array of viral infections are able to be contained due to the variety of T cells in the human body. The specificity of the adaptive immune response allows for a specialized, intense immune response to one pathogen. However, this specificity
He knows a lot about the science stuff. Will be examined in more.
Oh, I remember we watched this one. Yeah. You contradicted yourself a couple of times here, so let me untangle it. Viruses don't have DNA, they only have RNA. Actually, it's both. Some viruses have DNA, some have RNA. Never both, but it varies by virus. DNA viruses, herpes, smallpox, chickenpox. Interesting. RNA viruses, influenza, COVID, SARS, CoV-2, HIV, Ebola, measles. So there is no single rule. Each virus picks one nucleic acid as its genome. HIV is RNA virus, correct. And you're right that it was interesting from yesterday. HIV is special category called retrovirus. And your question about how it works is great. Here's the answer. Inject a cell into your DNA. HIV carries its genome as RNA. When it infects the cell, it brings along a special enzyme called reverse transcriptase that converts its RNA back into DNA. This is reverse because it goes RNA DNA, which is backwards from the central dogma. Is that CRISPR technology works same way then? So HIV virus has a special enzyme that it use called reverse transcriptase and can go backwards from central dogma. So it can produce DNA from RNA. My question is, CRISPR technology work exactly same way, or CRISPR technology actually been made from bacteria that stores chunk of DNA and able to cut it or attach chunk of DNA that it's been exposed to? I don't think I formed that question properly, but we'll see. That's reversed because once converted to DNA, another enzyme integrates, literally splices the viral DNA into your chromosome. It's become part of your genome. The cell now reads it as if it were its own gene, producing new HIV particles indefinitely. This is why HIV is so hard to cure. You can suppress it with drug, but you can't easily. You can suppress it with drug, but you can't easily remove DNA that's been stitched into your chromosomes. Your question about kit would it pass over? Your reasoning was exactly right. HIV integrates into the DNA of immune cells. Oh, into the DNA of immune cells, specifically CD4 T cell, not into sperm or egg cells. So it doesn't get passed to children. So through genetics, it can be transmitted during pregnancy, birth, or breastfeeding, but that's through viral particles and blood fluids, not through the inherited DNA. So it actually injects itself into T cells. Because my next question was like, can we develop a T cells that will kill infected cells? But who will kill the killer cell, right? Can a T cell kill another T cell? However, and this as well, the ancient retroviruses that did manage to infect egg or sperm cells millions of years ago, and their DNA is still in your genome today. About 8% of human DNA is fossilized retroviruses sequence. They called endogenome retroviruses. Oh, I think that's what it's interesting because like I have all those puzzles of information and they keep getting together. Like they become a one big picture of understanding. Most are broken and do nothing, but some have been reproduced by evolution to serve useful function. One example, a gene called syncytin, originally from a retrovirus, is now essential for placenta formation in pregnancy. A virus gives mammals the placenta. Really? No way. There is some mammals without placenta? You correctly identify HIV in array base, but incorrectly said all viruses are DNA. Reason. Running tally. Question 22. Hold on. Reverse transcript and HIV. Brief recap and anchor the comparison. HIV is a retrovirus in genome. Yeah. So, reverse transcript breaks the DNA RNA protein directionally by adding an RNA to DNA arrow. It's molecular copier that takes RNA as the input and produces DNA as the output. It doesn't cut DNA, doesn't target specific sequence, doesn't store memory of what it transcribed. It just copy whatever RNA it's handed into DNA. Got it. CRISP doesn't reverse transcribe anything. You have got the intuition right. It's derived from a bacterial adaptive immune system. So bacteria has immune system as well from viruses, and it keeps RNA or piece of RNA or some piece of information of virus on its own DNA. Okay, because bacteria also has immune system, not like our multicellular, but everything inside. Bacteria use it to defend against viruses. Yes, bacteriophages and plasmids. Here's how it actually works in its native context and how it was repurposed as a tool. CRISP is bacteria, the natural system. CRISPR in bacteria, in natural system, stands for clustered regulatory interspaced short palindromic repeat. Oh my god. I think in a day 69 I would be able to read it in one shot. 69, it's 63 days left. It's a region in bacterial genome that literally looks like repeat, spacer, repeat, spacer, repeat, spacer, where the repeats are identical TORI-BP sequences and the spacers are unique TORI-BP sequences. The spacers are snippets of viral DNA. The bacterium has previously encountered a molecular most wanted list. Love it. The system has 3 phases. Adaptation, memory formation. When a phage injects its DNA and the bacterium survives the attack, specialized Cas protein, Cas1, Cas2, grab a small piece of that invaded DNA and insert it Okay, let's go. So it grab a small piece of invade DNA and insert it into the CRISP array as a new spacer. This is the bacterium writing a mugshot into its genome. This is where the storing chunk of DNA part of your intuition is correct. Expression: the CRISPR array gets transcribed into long RNA, then processed into small CRISP RNA, rcRNAs. Each one contains single spacer sequence. Each gets loaded into Cas effector protein. Cas9 is a famous one, but there's many others: Cas12, 13, Cas8. So the Cas9 is a machine that's actually cut it, right? Interference defense. The Cas9 cRNA complex patrols the cell. When Cas9 encounter DNA matching its cRNA, it binds and cuts both standards of the DNA, a double strand break. The phage genome gets shredded, the infection is neutralized. Wow, the elegant part tells Cas9 what to cut, sequence-specific targeting, and Cas9 provides the molecular scissors. It's programmable by design. Swap the cRNA, change the target. Researchers realize if they can give Cas9 any guide at RNA we want, we can make it cut any DNA sequence we want. That's the whole inside the tool version is. But how we put it back? Complemented to your target DNA sequence, deliver Cas9 into cells via plasmid viral vector or ribonucleoprotein. Cas9 finds the machine matching sequence, cut both DNA strands. The cell own DNA repair machinery the cell own DNA repair machinery kicks in, and depending on which pathway activates, non-homologon and sloppy repair often introduce small insertion, deletion, gene knot. Homology repair, HDR. If you supply template DNA with the desired sequence, the cell use it as a repair template. Precise edit. Interesting. So there is a self repair machinery. DNA repair machinery exists in our cells, and if we supply chunks that actually fit into proper place, it will do HDR. If we don't supply it, then it just basically like cuts the knot. It's really programmable molecular scissors plus hijack the cell own repair system to install the change you want. Yes. The key comparison. Yeah. Okay, I need a bathroom break. Okay, change of plans. I need to pick up my daughter. I'll see you tomorrow.